Then I want to use a little bit of time to talk about molecular clock. Molecular clocks, this idea was proposed very early on when we start having the molecular data. At that time, it was like the protein sequence data. And Pauling and his colleague started thinking about if the molecule change at more or less a constant rate over time, then it would be helpful to use that to estimate the divergence time. So the idea why we call it a molecular clock is just the same idea as our clock on the wall or the watches on your hand. Those are they have like a specific scaling mechanisms to count every second and every minute in the same way. So it's the same in your watch and it's the same in the clock here. And it's the same in my computer here, although we cannot see it right now. But all the time are keeping or ticking at the same rate. It's like 1 second by the other. If the molecule evolve at a similar property, then we will be able to use the divergence or the differences between two molecules to understand how they evolved in the past and like how long have they been diverged. So most we consider the time. That's why we call it a clock. And when he proposed this, and he would like to consider if we can get all the sequences at that time, like a protein sequence, and we will be able to understand the rate of molecular evolution and to convert that to the divergence time. So that's the overall idea of the early times. And more or less that seems to agree with a lot of data. And a typical way to look at that is to have two independent measurements. So when I say two independent measurements is like one way, of course, we can look at amino acid replacement. That's like we collect the homologous sequence from a variety of individuals. But we like to test whether the divergence are sort of correlated with the real divergence time. Then we can use the organism with very good fossil record, because when we have the fossil record, we have a totally different way to estimate the time of this particular spaceman, and that is independent from our sequence analysis. So I want you to remember I use independent estimates here because in science we do this a lot. We want to ask one question and we want to ask whether this two has any correlation. Then we usually have to estimate the two things independently, use the totally different method. And this is what we did here. So in the y-axis is the replacement. Just like compare the protein sequence and look at how many differences occurred and estimate what would be the differences, what would be the changes happened in the past. So that's the y-axis. And the x-axis is the years. And it would be very nice we have all the different independent record for each lineage. Then we can plot them into this diagram and to ask whether we can see a linear pattern. If the pattern is linear, then that means at this molecule evolved at more or less a constant rate, and we can use that to estimate divergence time. So this is what we do. Although not every molecule can be used this way, but a lot of those conserved protein has been used. And we like to, as in the very beginning of this chapter, say we like to convert the substitution rate by year. That's this little r parameter we use starting from this chapter. And we like to know the changes per site per year. And you see those words in a lot of literature. Either they analyze the sequence with a group of organisms, and we know when they diverged. And we can use that as a guide to tell our reader, to tell everybody what's the substitution rate per site per year. Another approach we use is use a general idea from large group study, saying we know given the changes at this group of organisms as whatever number per site per year, we can estimate the divergence time of two organisms or any two organisms in this group. So this has a lot of assumptions, but the number we calculate really help us to understand the evolution of the organisms. So you observe that a lot of time, especially when we are interested in the evolution of organisms. When we reconstruct phylogeny, and quite often would discuss the origin of a certain lineage on Earth in the past, and we use the divergence time to indicate. And we also would discuss, in the discussion, we'll discuss back to that time what else happening on the planet. So this is how we do with all the divergence time and with the substitution rate. So the importance is here. Although there are different rates at different genes, and sometimes in the genome, not every site evolved at the same rate, we recognize that. So when we use that, we have to be really careful about what's the assumption we use, what's the guideline, which number you use, and whether everybody agree with the number. Of course, I say the number is great. With a number, we have some idea about back to certain million years what happened. But if you use the wrong number, it could give you very different results. So when you try to estimate divergence time, or when you try to show people what's the rate of evolution, you have to really carefully analyze your data and also search the literature, compare your results with others, and think about if there's differences, why, what type of evolutionary forces or mutation forces contribute to the differences. Okay, so all this I have been saying, like this is a very practical one, especially when we work on molecular phylogeny. So you can pretty much read these slides by yourself. And this one mostly is based on our neutral hypothesis. If a protein has very high selective constraint or evolve very rapidly, then you cannot really use it. Or put another way, they have to be, the locus has to evolve at a similar rate in different lineage. And that is not true all the time. So we want to show you, before we really do that, can we test that? Can we really test whether the gene evolved at a constant rate?